Molte novità sono in arrivo nella terapia del linfoma B a grandi cellule diffuso. Tra le nuove molecole, alcune sono in fase avanzata di sperimentazione, altre sono ancora in una fase un po' più precoce. Abbiamo parlato di questo importante e interessante argomento con il professor Greg Nowakowski. Professor Nowakowski, the therapeutic scenario of the lymphomas is growing day by day, we can say. So, which are the most important news in this moment, in your opinion?
Yes, absolutely. You're absolutely right. The portfolio of the drugs is growing, and we actually have two parallel developments in large cell lymphoma. One is the drugs which are agnostic to molecular signaling within the cells, but really focusing on surface molecules, and here are bispecific antibodies and antibody drug conjugates. A lot of them are already approved, and there are a lot of more in development. The other exciting developments are the small molecules, which are being developed as well. And we see them now coming and being integrated into this rapidly changing landscape of diffuse large B-cell lymphoma treatment. One of the recent developments have been that brentuximab vedotin, which is antibody drug conjugate, in combination with lenalidomide and rituximab in ENCHALON-3 study, showed actually improvement in overall survival when compared with rituximab and lenalidomide alone, or R2 alone. This is a late-breaking abstract at ASCO, so we'll see the exact details of the study. But nevertheless, from publicly released information, we know that the efficacy was much higher, and this higher efficacy resulted in a better overall survival in this setting. So brentuximab vedotin is not necessarily a new drug. It's been used in Hodgkin's lymphoma for a long time, also some of the T-cell lymphomas. But here we have it now as active agents in large cell lymphoma, probably because of the impact on microenvironment, in addition to directly targeting CD30 on tumor cells.
In this scenario, we have seen some data that are more promising than for specific new molecules. Can you tell something about this?
Yeah, absolutely. So for a long time we had immune modulatory drugs, and lenalidomide was a prime example. And lenalidomide is used in combination with tafasitamab in the treatment of relapsed refractory large cell lymphoma, is approved in this setting. And really lenalidomide combinations have been a backbone of many different treatments throughout lymphoma. Now, the exciting news is that we have a new generation of those drugs. Now they are called Celebron modifiers, and golcadomide is the new drug which has improved property in inhibition of Celebron, the target, and actually shows much higher clinical efficacy in the initial studies comparing with lenalidomide. Importantly, lenalidomide was mainly active in ABC subtype of large cell lymphoma. Golcadomide appears to be active across the subtypes and appears to be really potentiating also the activity of immune cells, effector cells. So it's really a good drug to combine with the antibody. Recently, we've seen at ASH present a study of combination of antibody with rituximab and golcadomide, and this combination resulted really in a high response rate of approaching 60%, and about half of those were CRs. So for the combination of the small molecule and antibody in the patients who are heavily pretreated with relapsed refractory large cell lymphoma, that's quite a significant activity. And the follow-up on this early study, phase 1 study, continues to see how durable those responses will be. But I do believe that this new compound will really change the approach and provide a new backbone for combination of the antibodies with bispecifics and combination with chemoimmunotherapy. In this regard, we also combine this golcadomide, or previously known as CC2H2, with R-CHOP, and we showed that it's actually very feasible, so you can combine it with R-CHOP. And this combination with R-CHOP now will be compared against R-CHOP alone in the randomized phase 3 study in the frontline setting, in the frontline treatment of patients with diffuse large B-cell lymphoma. So this is an example of the agents which was as a next generation based on the previous data, could really serve as a backbone to many new combinations, and it's really moving across the lines of therapy now to the frontline therapy. What I also was discussing that it can also be a good bridging strategy. A lot of patients who are relapsing with diffuse large B-cell lymphoma are candidates for CAR T cells, but frequently before the CAR T cells can be put in place and patients could be properly evaluated, the tumor can progress. So here, this is a perfect agent for combination with rituximab because it could stabilize the disease, it could actually shrink the disease and debulk the patients prior to CAR T cell therapy. And as of now, from preclinical studies and the clinical studies, it does not appear to be affecting the T cell numbers or T cell function, which is very important prior to apheresis for a CAR T cell therapy. So really a promising agent also as a bridging strategy.
Professor Nowakowski, there are some other molecules that are actually in study now. One of these is IRAK4, and I would like to ask you something about this new therapeutic agent.
Absolutely. IRAK4 inhibitor emavusertib is very promising in this setting. We know for a long time that IRAK4 signaling in large B-cell lymphoma is very important, particularly in ABC subtype or MCD subtype with MYD88 mutation. And we now have studies which combine emavusertib with ibrutinib. And indeed, this combination results in responses in patients who are actually resistant to ibrutinib or BTK inhibitors otherwise. So here you have a proof of concept that patients who are progressing on BTK inhibitors can actually respond to the combination of this dual inhibitor. And we have seen some responses in patients who are heavily pretreated with primary CNS lymphoma. And that's a major strategy right now for development of IRAK4 inhibitor. And this is a huge unmet need and extremely promising really approach to treatment of those patients.